Greetings to you all, dear students. Today we begin with another unit in the course Statistics for Engineers, and the name of the unit reads Design of Experiments. I'm bringing in a series of videos again in this chapter for your later reading, and this is an introductory part of this particular chapter. So let's begin. As the title reads Design of Experiments, it's important for us to know how we define an experiment. As you all can guess, an experiment is performed to discover something about a particular process or a system. It's in fact a test that we conduct or a series of tests which tell us what are the changes that can happen in the output variables when there are changes made in the input variables. And the identification of such possible changes is what we detect in through an experiment. So in short, an experiment is conducted with an objective of verifying a certain hypothesis. Now the term hypothesis only means a statement that is made based on a particular evidence or a reference, and it is this experiment which will prove whether the statement that we have made based on the existing evidence is either accepted or is it rejected. So hypothesis is a statement which is being verified through an experiment. This is one way of defining an experiment. The other way is when there are input variables given to a process or a system and we make changes in it, then it is obvious that there will be possible changes in the output variable, and that kind of a relationship is what this experiment will tell us about. In statistics, these input variables are termed as factors or treatments, and the response that we get and the output variable that we get, we call it to be a response variable. In fact, these words are used because it has been this designing of an experiment is initiated originally by the Britisher Sir Ronald Fisher, and he had conducted in the presence of many naturally fluctuating conditions how valid experiments could be conducted. So based on his theory has this design of experiments come up. Today, we not only see it very widely used in agricultural research, but in any field that we can think of. Let it be biology, let it be psychology, or even today data science. So, in many fields where we need to develop a relationship between the input and the output variables, that is, factors and responses, we apply the method of design of experiments. And as I said, R.A. Fisher is credited for his work in design of experiments, which in short is a valid abbreviation as DOE. Let us look at the variables which exist in any conducting an experiment. So when I conduct an experiment, I may have a system or a process and certain input variables that I am placing, which call it as factors, and then we obviously get some output variables based on the process or the system that we have, and we give it the word as a response. Now, I'm sure you all agree that it is not just that the input variables affect the output variables. There are number of other fluctuating variables that act on a system. So those variables which are directly involved in the experiment, such as the input variables directly attacking the system to give you the output variables, are called experimental variables. So variables that are directly involved is called experimental variables. And those variables, I also told you that there are variables that can act on the system which may also deviate or fluctuate the output result. Instead, so our focus is only on the input variables to get the output variable, but there are other factors which may affect the system and thereby the output variables. So our interest is only on the experimental variables which directly attack the system, whereas these variables which also are a part of the system and which cannot be avoided are called extraneous variables. So variables that are not directly involved but have some effect on the experiment are called extraneous variables. Now these are the variables which can give you an inaccurate conclusion, and thereby it is important that we minimize the effect of these extraneous variables. So this is one of the objectives of design of experiments. For a simple example, if I take up, I say the hypothesis or the statement that I get through an evidence is that manure increases the yield of paddy. Now, this statement is to be verified through an experiment, but here we are only now defining variables at this particular level. So, the experimental variable is nothing but the quality of manure, and the response is the quality of the paddy field. And what are the other factors that can affect that particular yield would be the soil fertility, amount of rainfall, quantity of seed, quality of seed, and the quantity of seed that we put in, and also the moisture, soil moisture. So when I talk about so many effects, all these are affecting the yield of paddy. It's not just the manure. So if I focus only on manure, then it is only this is the input variable, this is the output variable. But these variables also act on the soil, and therefore we have to reduce the minimize the effect of these in order to get a proper conclusion between manure and yield. Such variables which are not directly related are called extraneous variables. And our motto in design of experiments is to minimize the effect of these extraneous variables which may deviate the conclusion of that particular experiment. So let's look at the objectives further in the next slide. So before I move on to the objective, here are a few terms which are important for our understanding. First is called the comparative experiment. Naturally, as the word says, the experiments are designed to compare the effect of two or more objects. When I say objects, it could be treatments on some population characteristics. The population, when I say it, need not be individuals or people, it could be even plots if I am talking about an agricultural situation. So the experiments that are designed to compare any two of the objects, like say fertilizer A, fertilizer B, which is applied to certain plots, and then that would become a comparative experiment. So as I said in an example here, comparison of different fertilizers, different varieties of crop, different cultivation methods. So when everywhere we are trying to compare two different quantities, objects. So those are called comparative experiments. Treatments, as I told you in my previous slide, different objects of comparison like these, different fertilizers, varieties, methods, they are all objects and they are called treatments. So different objects of comparison in a comparative experiment are called treatments. So it could be different fertilizers, different varieties of crops, etc. What is an experimental unit? A unit on which an experiment is performed or treatments are applied is called an experimental unit. For an example, when I talk about treatment of fertilizers, naturally the unit or the place where the experiment is performed is a field. So I call it to be plots. So plot of land in field experiments is an experimental unit. So the unit on which the experiment is performed is called an experimental unit. Now, there are situations where I may be having, say, I have two plots just beside one another. I give in a treatment A here, and here also I may give the same treatment A, and but the yield that I get out of this is different. The two yields are different, though the region or they lie in the same region. So there are a lot of factors that affect the result of the yield, though the plots are just beside each other. It could be the soil fertility at that particular point, or the moisture levels of this in each area. So this kind of a difference that is noticed in the output variables or in the response variable creates what is called an experimental error. So the variation that we get from in the results that we see from plot to plot is called an experimental error. There are number of factors that are related to this. We cannot say this is the reason why there is an error. So the factors are plenty, and our motto will be to reduce the experimental error. Now that we have understood these definitions, we are now in a position to state the objective of why is it that we are learning this designing or planning of an experiment even before we perform it. Let's see what it is. So, having seen a little of theory in our previous slides, now let us define what is the objective of DOE. Now, the objective that we set is two main things: to control extraneous variables. So, the variables that are not relating to the experiment directly but indirectly is affecting the response variable. So, what we mean by saying to control, it means to minimize, to restrict. We cannot completely remove them off because they exist in nature, and it is an ideal situation where we can remove these extraneous variables. Since we are not talking about an ideal case, our motto should only be to control it. As is always said, any real problem that we do does not give us 100% accuracy. So there is some amount of error that is possibly and that can be accepted in for our further work. So our motto shall not be to completely eradicate them, but to control them or to minimize them. And the second condition objective is to minimize the experimental error. So this is also in short the response of these extraneous variables. So in short, the DOE is mainly used to reduce the error component and reduce the control of when I say reduce, I should reduce the effect of the erroneous variables, right? Extraneous variables, or you can call it as the experimental error to be minimized. Coming to the basic principles, now we want to control these extraneous variables, we want to minimize the experimental error. How can these be done? So Fisher is the one who said there are certain ways in which we can restrict the effect of these variables which are not needed and thereby reduce the error. The first of it is called randomization. So what does randomization mean is apply in a random fashion. So we have a lot of softwares today which create random numbers, which generate random numbers. So likewise, when I am placing or the technique of identifying or allocating a treatment to an experimental unit, I do it in a complete random fashion. That is, I don't put it as A, B, C, D. That's not the order I would follow. But there are a lot of softwares and a lot of techniques in order to prove this randomness. How do we do it? One simple example is, suppose I am I have three treatments here, and I have four plots wherein I have to apply the three treatments. Now, what I can do is I can take three cards, write A on one, B on the second, and C on the third, shuffle them well, and then when I want to identify which treatment to be given for plot 1, I pull out a card. I notice that A is written on the card, so I apply treatment A on the first card. Similarly, I put back the card into the pack. Again I shuffle and I pull out one more card. for filling up the second plot, and I see that it is C. I put it back and put again shuffle, pull out the card. So I see here that treatment A is applied for two plots here, and one is for B and one is for C. So this had no particular reason of choosing ACAB as the combination. It was pure a random manner in which we selected the treatments for these plots. So randomness is a very important tool which will remove bias in the experiment. So there is nothing called favoritism. There is nothing called, this particular plot I need to put it as A, this particular plot I need to put it as B. So nothing is fixed beforehand, and it removes this concept of bias, which makes the experiment more transparent and viable. Let's look at the other principles now. Now, the second principle, the most important and interesting one, is replication. When I talk about replication, in English it means repetition. Why do I want to repeat something again and again? Naturally, you would say that when I repeat an experiment, I expect a same result. When I'm doing a problem and I repeat the problem, I expect the same answer. So repetition here is only ensuring consistency. Consistency, and on an average, when you repeat, maybe slight differences when you differentiate, when you replicate an experiment, and you can take an average to recognize the influence of the factors on the experimental units. For an example, I take up 12 plots and I have three treatments. Again, I assign them randomly, as I explained to you earlier, and I repeat the process in order to see that the result I get is consistent any number of times I repeat it. This proves that the experiment that I'm performing is on normal lines and there is no mistake. In case we get a lot of variation between the results in two or three attempts, that means the initial basis for setting up the experiment itself is wrong. So, randomness removes bias, replication ensures consistency, and let's look at two more. The third principle of DOE is local control. As we have already stated in our previous slide, our motto is to reduce the effect of the extraneous variables. And one way of doing it is blocking. What does blocking do? Let me explain it by an example. I take a drug, I administer it to certain individuals and to check on their weight loss. Is there a weight loss when I use this drug or no? Or is the weight gains? So my question is, does this drug ensure weight loss? So what am I doing? I'm taking certain individuals, say some male and female individuals, and I administer the drug to them, and then I check on their weights before and after administering the drug, and I get an analysis and the conclusion is made. But there is one point to note here. The way a male responds to this drug is certainly different from how a female responds to the same drug. So I have not considered that aspect here and just taken them to be individuals, and I have checked on whether the drug is really decreasing their weight or not. So this particular main aspect of the sex of the patient is not considered here. So what I can do is I realize that this is a factor which can really adversely give me a wrong conclusion, and therefore I need to block this extraneous variable, which is the sex of the patient. As I said, I cannot block it completely, I cannot eradicate it, but I can do one way. So look at this graph given here, this block diagram. Individuals are taken. I randomly divide them into two groups. So there's a group containing male and female here, male and female here. I give this group a particular drug, and this group I don't give them anything. That is, I control the variable, I don't give them anything here, and then I check on the weights after the time duration and conclude my answer. But this is again a case where I am not separating out male and female. And I realize that that could be affecting my conclusion. So I use the concept of blocking. So what do I do? The individuals here, first of all, are divided into two blocks. One which will be male members and the other which is female members. So block 1 is completely male, say, block 2 is completely female, and then each block, as we did earlier, is randomly divided into two groups. Female again is randomly divided into two groups, and I give one group the drug and the other group no drug, and then generalize. Then the result that I get will certainly be different from case 1, because block 1 results will be pertaining to how men respond, and block 2 results will be pertaining to how females respond. So this conclusion is much better than the case when blocking is not done. And this blocking plays a vital role in controlling the effect of the extraneous variables. So therefore, there are three main principles: randomization, replication, local control, and under local control, blocking is our technique which will control these extraneous variables to a greater extent. We have now studied the basic introduction of DOE. Why is it created? What is the objective? And what are the principles of DOE? Now we shall take up some basic designs in this particular chapter: completely randomized design, randomized block design, and Latin square design. These are the three designs which we shall elaborately study, and then we shall solve these using analysis of variance. This is a very important tool in statistics to check the variations in the output. ANOVA, in short, it is called. It's a technique used for analyzing variances between two samples. It enables testing significant difference between variances of samples, and it uses the F test for making inferences on the variability between two samples. Kindly note that this variability has to be reduced. We do not want to see much of variability in our experiment. So all our efforts are trying to minimize variability and improve consistency, improve the accuracy of the result, control the effect of extraneous variables, and minimize the experimental error. So the entire design that we are going to deal with, design of experiments in all the three models that we are going to deal with, focuses on these aspects. So in my next video, I shall define what do we mean by a completely randomized design. Then let us study how ANOVA helps us in analyzing that design and by giving us proper conclusions using the F test. So thank you for listening to the introduction. I hope you all have understood it, and you can even ask me your doubts in case you have any. So let us meet in our next video or with the completely randomized design topic. Thank you. Good wishes.